Our blood is more than just a transporter of oxygen or nutrients. It's a rich, dynamic map of our internal chemistry, one that researchers are now learning to read with extraordinary precision. At the forefront of this discovery is the study of metabolites, small molecules produced during metabolism that reflect the biochemical processes constantly at work in our bodies. From amino acids to lipids to organic acids and sugars, these metabolites are emerging as powerful indicators of future health. By analyzing them, scientists can often detect disease risk long before symptoms appear, opening the door to a more predictive, preventive approach to medicine. Unlike traditional tests that focus on a few isolated parameters like cholesterol or blood glucose, metabolomics, the comprehensive study of these small molecules, offers a panoramic view of health. It captures the interplay between diet, lifestyle, genetics, and environmental exposures in real time. In doing so, it allows researchers and clinicians to identify metabolic fingerprints associated with the early stages of chronic diseases such as type 2 diabetes, cardiovascular disease, and neurodegenerative conditions like Alzheimer's. For example, researchers have found that certain amino acids, such as branched-chain amino acids, BCAAs, are often elevated in individuals who later go on to develop insulin resistance and type 2 diabetes. This elevation can be detected years before fasting glucose levels begin to rise. Similarly, certain lipid metabolites, including specific types of ceramides and acylcarnitines, have been associated with higher risk of heart attacks, even in people with normal cholesterol levels. These markers seem to signal subtle disturbances in mitochondrial function and fat metabolism that precede clinical cardiovascular disease. In the realm of brain health, the metabolomic profile can be equally revealing. Elevated levels of homocysteine, combined with changes in phospholipids and oxidative stress markers, are increasingly being linked to cognitive decline and dementia. These changes often appear well before any cognitive symptoms arise, suggesting that metabolic imbalances may be quietly reshaping brain biology in the background. Importantly, these imbalances are not always irreversible. In many cases, dietary interventions, exercise, and micronutrient optimization can correct them, potentially delaying or even preventing disease onset. The real breakthrough lies in the predictive power of these markers. Instead of waiting for a diagnosis based on outward symptoms, we can now begin to act on early biochemical warning signs. For instance, in large population studies, researchers are using metabolomic profiling to classify people into risk categories for conditions like hypertension, stroke, and metabolic syndrome, often more accurately than with traditional risk factors alone. This allows for earlier, more personalized interventions tailored to an individual's specific metabolic vulnerabilities. What makes metabolites especially valuable is their responsiveness to change. Unlike fixed genetic variants, metabolic markers fluctuate in response to what we eat, how we move, how much we sleep, and how stressed we are. This dynamic quality not only allows them to predict disease risk, but also to measure progress in real time. If someone adopts a healthier lifestyle, such as shifting from a processed Western diet to a Mediterranean one, their metabolomic profile begins to shift within days. Improvements in lipid profiles, reductions in inflammation-related compounds, and enhanced energy metabolism are all visible in the data, long before outward changes like weight loss occur. Some researchers envision a future in which routine blood draws generate personalized metabolic health scores that guide diet, exercise, and supplementation with precision. Already, some clinics and research centers are using metabolomic panels to guide therapy for individuals at risk of chronic conditions. It's a new paradigm for healthcare, one grounded not in waiting for illness, but in detecting and correcting the earliest signs of imbalance, quietly written into our biochemistry. The growing field of multiomics, which integrates metabolomics with genomics, proteomics, and epigenomics, adds even more depth to this approach. It allows scientists to connect metabolic disturbances to genetic susceptibilities, protein-level dysfunctions, and gene regulation patterns. The result is a more complete, individualized picture of health and disease risk. Studies like CIO, which track these various layers in long-living populations, are helping to identify the biological pathways most closely tied to exceptional health span. In these populations, the metabolomic signature is often characterized by low inflammation, efficient energy metabolism, and strong antioxidant defense, factors that seem to buffer against aging and disease. Perhaps most empowering is the idea that our metabolic future is not fixed. The body constantly responds to input, and the metabolites we carry reflect our habits, our environment, and even our emotions. Each meal, each night of sleep, each walk, each stressful conversation leaves a biochemical trace. By paying attention to those signals and using them not just for diagnosis but for direction, we may be able to steer ourselves toward a longer, healthier life, one informed not just by symptoms but by silent, molecular whispers that speak of resilience, balance, and possibility.